Multispan is a San Francisco Bay Area-based company. Our goal is to enable our customers to make GPCR drug discovery much faster by providing sensitive, robust, and diverse assays. If you would like to discuss with us how Multispan can provide you with GPCR reagents tailored to your specific needs, our contact information is listed here. You are also welcome to visit our website at multispaninc.com and follow us on our brand new Twitter account, twitter.com/multispangpcr. Here is a list of products and services that Multispan currently offers. This list includes the following cell line products: an extensive catalog of GPCR stable cell line clones, selected and validated for pharmacological functions, division-arrested frozen cells, and frozen cells for membrane preparation. Multispan also offers the following services: custom GPCR cell line and assay development, GPCR compound profiling and screening in agonist, antagonist, and modulator modes, and antibody binding and function profiling. Multispan has established an extensive platform for drug discovery compound screening and lead optimization, bringing the industry's largest collection of GPCR cDNAs of 320 human full-length GPCRs and 180 rodent GPCRs into Multispan's proprietary expression technology. Using our assay development expertise, we have established 250 individual validated cell-based assays and a 75 target assay panel for orphan GPCRs. Many GPCRs are known to be difficult for assay development due to 1. A lack of sufficient surface expression, or 2. Desensitization by endogenous ligands, or a combination of both. But scientists at Multispan have overcome these difficulties and have successfully generated stable cell lines and developed assays for many of these difficult targets. These targets include cannabinoid, chemokine, free fatty acid, lysophospholipid, melatonin, metabotropic glutamate, muscarinic, neuropeptide, opioid, prostanoid, purinergic, and somatostatin families. And our platform is compatible with multiple assay formats, such as molecular devices Flipper Calcium 4, Sysbio's HTRF cyclic AMP and IP1, Promega's Glow Sensor cyclic AMP, and reporter gene assays. Multispan has a broad panel of full-length human GPCR cDNAs that already spans 63 different families. The blue bars represent the number of GPCR targets already cloned into our proprietary vector, whereas the red bars illustrate cDNAs being cloned. Multispan also has a broad offering of functional assays that already covers 59 different families. The blue bars indicate assays already developed, while the red bars show assays currently under development. Our rigorous cell line generation and, more importantly, quality control process ensure robust and high-quality cell lines that are delivered with a short turnaround time, typically 60 to 90 days. Each single cell clone is selected for a specific pharmacological function of the expressed GPCR target and will undergo 2 months of stability testing. In addition to 1. Multiple functional validations, 2. Receptor verification by RT-PCR and sequencing, 3. Mycoplasma testing by 2 different methods, and 4. Cell growth curve testing to make sure that the cells at least double every 24 hours. Before animal studies are carried out, it is important to test compound leads in pairs of cell lines that are expressing similar levels of human and rat or mouse GPCR targets, and that demonstrate similar receptor pharmacological functions, to avoid using expensive animal models with very different receptor pharmacologies or misinterpreting data from such animal studies. Here is an example of 3 cell lines developed at Multispan, each expressing human, rat, or mouse GPR120 target. Comparable functions of each receptor is demonstrated with linolenic acid and GW9508 compounds, whereas comparable surface expressions are shown by FACS. Many of our cell lines are suitable for multiple cellular functional assays, so that we and our customers can take advantage of utilizing different assay readouts in different stages of a screen. As an example, many of the mGluR family receptors are coupled via G-alpha I, which signals through the cyclic AMP pathway. We developed both cyclic AMP and calcium assays for G-alpha I coupled mGluRs. One can take advantage of the large assay windows of calcium assays in primary screening, and then use the endogenous pharmacology of cyclic AMP assays in follow-up studies. A summary of all the available mGluR assays is listed in the table. mGluR2 cell line and assay development is shown in this slide and the next as an example of assay diversity. One cell line with two cellular assays. Upon activation with the L-glutamate ligand, mGluR2 is shown to inhibit forskullin-induced cyclic AMP in a dose-dependent manner in the two graphs on the left. The two graphs on the right show an activation of calcium levels in a dose-dependent manner with increasing levels of the ligand. The activity of the mGluR2 agonist modulator, BINA, can enhance agonist-induced receptor activity, as measured by the cyclic AMP assay on the left and the calcium assay on the right, which is shown in color purple. In addition to agonist and antagonist assays, this cell line can also be used to screen positive modulators in either calcium or cyclic AMP assays. Here is a typical example of a compound profiling experiment that we perform for our customers at Multispan. The compounds are initially analyzed in a single dose across 30 assays. Compounds that show specific agonist activities for rat and human NPBW1 and human opioid delta are then further analyzed in dose-response curves using serial dilutions across 8 concentrations. Identifying harmful off-target effects early in the functional assays allows scientists to save significant downstream drug discovery cost and time. Multispan focuses rigorously on the quality of our services and the integrity of our science. We take great pride in our high customer satisfaction and customer return rates. Here is feedback from a few of our customers. As a company, Multispan will continue striving to bring products and services for novel GPCR targets with highest standards to our customers. Thank you. Again, we can be contacted at sales@multispaninc.com or by phone at 510-887-0817. Please also find out more about us at multispaninc.com and twitter.com/multispangpcr.